thanks for the introduction, kind of saved me some time. Today we're going to talk about genome secondary structure, and it's nothing new. So that's like the first thing to say about it, but we'll get to that. Also, the presentation with all the links, with all the sources, references, even the code, all the graph, everything is publicly available. Either scan this or just search my name, you'll get to it. So RNA virus discovery is exploding if you haven't paid attention or if it's not your field. And I was lucky enough to be involved in one of these projects, as Simon said. And I hope that we will hear about another project like that today, later in Eddie's talk, but I can only hope. So as more and more viruses are being discovered, and I'm specifically talking about today RNA viruses, we just understand very little more about them, not as much as the sequence get accumulated. Now, most discoveries rely on either sequence or to some extent structure similarity to known viruses. And from that, they can infer phylogeny, the protein, what do they catalyze, what is the structure importance, and ideally, who is the host in ecology. So just from a bird's eye view, one of these expansion projects was actually done just here using the JGI data, the IMG data. I'm quite happy and very humbled to see some of the actual data generators right here in the crowd. And I'm not going to talk about it too much because in a large regard, this is last year's result. This is last year's snow. So basically, we found a lot of new viruses. And for what we could, we tried to explain what do they do, what's their ecology, who's their host. But that only led us so far. So this source for this graph is just my gut feeling. This is based on nothing concrete, but we are somewhere on the beginning of the sigmoid curve of explosion of more and more new viruses. But what we can say about them is not growing at the same rate. We can do a lot of stuff about a handful of viruses. There are more potato viruses than you would think. And there are much more diversity than just potatoes out there. Most life is microbial, yet we are only trying to see the cusp of microbial RNA viruses. So for most of them, we basically know nothing. We know that there are viruses, probably, hopefully, but we do not know the genome polarity, whether or not they're single-stranded, double-stranded. We do not know if they have multiple molecules encoding the genome or just the one with just the gene that we were able to identify. We have, for some of them, we can say, based on protein similarity, how do they look in 3D? Are they, icosahedral like we like, or like my shirt says, or how they helical, rod-shaped envelope? Do they even have a capsid, which is a thing? How do they packaged? And we don't even know if they are complete genome. If we take them to the lab and try to do symbio with them, will they even be able to create infectious progeny? And the most important thing there is, in biology, there are a lot of exceptions. There are no obsolete rules, but there's only one thing that such rule that I've ever found so far, which is that all viruses are obligate parasites. And for the vast majority of them, we do not know the host. So shame on us, let's do better. Okay, so genome structure, and I don't know how much time I even have left, but genome structure, a lot of us think about genome, computer scientists will think about it as a string, essentially an array of letters. But realistically, this isn't a string, it's more like pasta or ramen. It's folding over itself and it's not consistent. In certain temperature, it's like that, in other temperatures like that, in certain times of the infectious cycle, it's completely different than otherwise. And nothing new, the first genome ever sequenced, incidentally an RNA bacteriophage, how important is they had a 2-page figure just with the genome structure. Now, why is it so important for RNA viruses? They can't really control when the DNA, which they do not have, becomes RNA, becomes active transcript. So a lot of not for all of them, they have a lot of tricky machineries, whatever, but many of them use these as trick to control temporally when do more or less of different proteins get expressed. So if you think about it, some of them would, you have 178 units of the CP protein in the MS2 virus, for example, for one RDRP, one of their polymerases. So how do they do it if they're all cistronic on one mRNA opera? Okay. They do use it for other stuff, which I won't get into it. And it's, again, nothing new. We know it from all the way up to COVID. There's important structures there, and we know it since the start. Okay, so hypothesis time. If different viruses use different RNA secondary structure, we should be able to measure it if we're competent enough, and we should be able to see distribution differences across the different types of structure. And if we're really lucky and really competent, we might even be able to build classifier based on those information and try to say something about all these new hurdles of viruses that we can't. So spoiler, we successfully failed for the genome polarity. And what I mean by successfully failed is that by failing, we realized that we see no signal, no obvious trend just for genome polarity. No, apparently there's no universal marker for whether or not they're single cell and double cell or whatever. But what we were able to see is that that's because what we would have considered noise with this target variable is actually very, very important features for the taxa. So essentially, different lineages of viruses use very, very specialized combinations of secondary structure. So if we actually change the target variable back to what is the lineage without even doing any protein analysis, without even predicting where the proteins are from start to stop, just looking at the structure, we can pretty accurately say, or better than a coin flip at this point, what is the virus taxa. So I won't get too deep into the actual bioinformatics pipeline. It's all open source. You can Google it. But we've took the information from the project I was involved in 2022. And we basically picked a few groups that only from the lineage are consistently either have this genome polarity and with some, this is, by the way, this is all Jupyter notebooks and they're Google Colab compatible, so you can run it on your own data or check that I'm wrong. So essentially, we basically quantify the different structure, the number of hairpins, interior loops, whatever, and then we transform them into frequencies, how many per 1 kilobase pair. Now this is colored based on the genome polarity, and without looking too deep into and each row is a different family, not just mostly families, one of them is an order and one of them is a kingdom. But what you can see is that there's no obvious trend. There's not like the distribution is highly skewed if you group it or aggregate it by color. But when you look at that, this is kind of like a chicken and egg when I already show you this by breakdown by taxa, and you can see that there is a quite interesting distribution. Now, these are two features, the percent of unpaired bases and the minimum free energy that are not directly structure shaped, but the structures themselves, the frequencies of the structure follow the same kind of weird distributions that I'm going to skip a bit, but there are a bit of interesting statistical insights we can get from them, but most of them are banal. For example, longer sequences would have more elements. Cool. The variance is significantly not equal. Also, good. So very, very briefly, this is visualized using Orange. If you are not very competent or sure of yourself with coding, you can do amazing stuff with it for data mining and data analysis. So that's just the word. Including even doing machine learning stuff, although check yourself later. Anyway, basically, this is the simplistic split train test and then tenfold cross validation. If this is not English for you, it doesn't matter too much. Basically trying to fit a model or fit a machine learning algorithm that could predict whatever variable we try. So we can change which variables are the input and which variables will be the target. And that would change, by the way, which model we need to select. After that, we test the different models, and then we try to see which features were more important. So without going too much into it, here are the raw data, which, again, I don't have a lot of time. But it's, if I could just summarize it, it's way better than a coin flip, but not really that good. And the main reason for that, I guess, is that we still haven't gotten to do any feature prioritization. This is just, so far, has been kind of like an afternoon project for me. And I hope we could use this, maybe not for taxa delineation, but use this to try and do something better, because this is not dependent on protein similarity to known stuff, which is where we've been paddling all this time. So some conclusions and limitation. RNA secondary structure appears to be lineage specific, which is good and bad, means that it's, again, we have overrepresentation of potato viruses for good and for bad. We might be able to use this to assess completeness as well, because so far what I've showed you is actually not really normalized, but transformed to frequencies. If we look at the absolute count and we have representative that we believe to be complete, we could use that as well. This is inaccurate. So the RNA folding that I've used is, it runs fast, but it's at best 80% accurate, and it's been trained and optimized for E. coli at 37 degrees, or human serum, or structures in PBS. We are looking at stuff from geysers and the bottom of the ocean, and wherever in the world. You would know way better than me. And this is not really the conditions there. There's also a lot of structures that we just cannot predict accurately, like G quadruplets and stuff like that. And we probably observe an overrepresentation of the main marker gene of RNA viruses we try to do. So we still have a lot of way to go. Now, I want to acknowledge both my mentor and PI for the last more than a couple of years, Uri back from Tel Aviv University, but also the extremely talented undergrad who did the study or did a lot of the prototyping, who has the resilience to see that it fails and yet not realize that it still has value. Rima Sajjal, and all of the authors and the data generators from the RNA virus meta-transcriptome project, the AlvinP project, Danielle Viraz and Rhys German from San Diego State for really helpful discussion, and the Safra Center for sending me here. Thank you.
Thank you so much. Questions? Oh, one in the chat as well. I didn't have it in the chat. Okay. Oh, that's from the previous talk, right? Yeah, microburst plate system. Okay, that's a question from Vanessa. So Vanessa, you will have to stop by, or like a social learning event question. Let's do a Neri question. And I can start us if we need to. Okay. You mentioned multi-segmented viruses. So viruses with like their genomes in multiple pieces. Do you find any signal that like predicted structure could help us identify which segments works with which other segments in a complex community?
That would be one way to use this kind of approach. Some families of viruses use basically the packaging signal is either secondary structure or primary structure. So even the order in which they get encapsulated, which segment gets into the protocapsid first is also a thing that is governed by that. So yeah, there might be something there.
Okay, so what I hear is there is hope. I like it.